All right, good morning, everyone. So I'm going to be talking about some ongoing work in the Materia lab looking at the genetic basis of female mate preference in populations of Drosophila melanogaster. So when we think about speciation, one of the most important aspects of this is reproductive isolation, a mechanism through which species stop mating and then are no longer able to form fertile offspring. And in this talk, I'm going to be focusing on pre-mating isolation, in which the organisms simply do not mate. If you look in the literature, this is generally considered to be extremely important for speciation. That sort of makes sense. If species can no longer mate, gene flow ceases, populations will begin to diverge. Additionally, we know from some work that Coyne and North have done that pre-mating isolation evolves substantially more quickly than post-mating isolation, suggesting again that this is important to the overall process of speciation. Despite its apparent importance, very little is actually known about the genetic basis of pre-mating isolation in different populations. But you'll find that a number of systems have been described. And the system we'll be focusing on today is the cosmopolitan Zimbabwe matrix system in Drosophila melanogaster. So this system was initially discovered in the mid-90s. A collection of flies from northern Zimbabwe and southern Zambia, when sequenced in the US, found a very high level of divergence compared to the cosmopolitan melanogaster found throughout the rest of the world. Even very cosmopolitan melanogaster living on very different parts of the globe were more similar to each other than to these flies they found in Zimbabwe. And the authors guessed that, or found that these flies probably started to diverge about 10 to 15,000 years ago. Wu and colleagues, when looking closer, found that this is due to extreme female preference in these flies. So the cosmopolitan melanogaster females will choose males from their own cosmopolitan population, not from the Zimbabwe population, about 2 to 1 when given a choice between two males. Females from Zimbabwe will overwhelmingly choose males from Zimbabwe. They have almost no interest in meeting with the cosmopolitan males. And we recapitulated this in our lab and a number of other labs as well. We find this consistent that females from these Zimbabwe lines overwhelmingly prefer Zimbabwe males when given the choice. Holliker and colleagues following up on this work wanted to map what part of the genome was involved in this mate choice. In 1997, they found that if you do chromosome substitutions in females, so basically these are chromosomes with one Zimbabwe chromosome and 2 cosmopolitan chromosomes, chromosomes 2 and 3 are by far the most strongly associated with this trait. And some data that I won't show here from our lab found that if you introgress different chromosome arms of Z into an M background, 3R is the most strongly associated with this phenotype, so I'll be focusing on that later in the talk. Something that's very important that I need to stress is that Zenus is recessive. So when you make F1s between cosmopolitan and Zimbabwe flies, they behave like cosmopolitan 100% of the time. This has been found in every lab that's ever looked at this. This is quite important for what we're going to do because we can take advantage of the recessiveness of the Z character to use deficiency mapping to find what part of the genome is involved. So what we did is take deficiency stocks, which are, you can get these from Bloomington, there's some deficiency over balance of chromosome, and tiled this across the whole of 3R. You take these flies and you mate them to a Zimbabwe fly, which is, homozygous Zimbabwe, and you'll get 2 types of progeny. One that is a balancer chromosome over a Zimbabwe chromosome, and one that is a deficiency chromosome over a Zimbabwe chromosome. Now, these flies will behave like melanogaster flies always, and these flies will behave like cosmopolitan flies everywhere except where this deficiency has uncovered the recessive Z allele. And so if you see these flies, if you see these flies behaving like Z flies, you know that something in that deficiency is affecting the mate choice. The way you do these experiments is quite simple. You take a female that's either a deficiency over Z or a balancer over Z, put her in a vial of food with 2 males, one cosmopolitan male and one Zimbabwe male. And you feed these males colored food so that you can differentiate between them. We and a number of other groups have published on that. The food color doesn't make a difference. The females don't care. So that's a great way to distinguish between the males and try to figure out which she's mated with. You let them go for a little bit. Eventually, the female will choose a male and mate with him. The other male will be neglected and sad. You aspirate him out of the vial, and you score the number of males that were chosen for each of the phenotypes that you tested. So you get data that looks a little bit like this. So this is the deficiency of this will be this cytological band. You can see these balancer flies behaving like N flies, as we expect. slightly preferring the M flies, but not the enormous preference you see in a Z fly. And when you do the deficiency on this region of the chromosome, you see no real change. So whatever we uncovered there doesn't seem to be involved in mate choice. This stock, on the other hand, so again, we see balancer over Z behaving like M. But the deficiency stocks suddenly start behaving really quite like Z flies. These flies have a very marked preference for the Z males. And we see this with two other stocks as well. They are M flies when they're balancer and Z flies when they're deficiency for both of these cytological bands. So for all three of these, we found something in a region between them that is causing Z-like behavior. And all of these associations are quite statistically significant. So when you actually look at these on a chromosomal map, the region they map down to turns out to be about 150,000 base pairs in length. And there are three genes of interest in this region: Kinkar, couch potato, and Grin. So that gave us three drug candidates to start looking at. So when you look at Kinkar, Kinkar is involved in photoreceptor cell development in the eye of the fly. So intuitively, that wouldn't be the strangest thing to find involved in mate choice. But when you make P element insertion null stalks and do the mate choice trials, these flies behave like M flies. They have the preference of M flies, they don't care. So Kinkar is not involved. Do the same thing with couch potato, and once again, when you do the P element insertion nulls, they just behave like M flies. You don't see the same result you saw with those deficiencies. That very, very strong marked Z preference. Rim, on the other hand, you do see it. Rim is involved in synaptic transmission and plasticity in the central nervous system. And when you make these P element insertion nulls and do the matrix experiment, they behave like the deficiency flies. So rim encodes for a protein called radically interacting molecule. It is involved in synaptic transmission and synaptic plasticity, specifically vesicle binding and release. And here's something that's great. There are two very highly integrated sites between the M and Z alleles of the rim. So there's one site that is a G to A transition. That's at about 80% allele frequency difference between the M and Z populations, and one that is a C to T at over a 50% allele frequency difference between these populations. So now we've found that rim null flies recapitulate this phenotype, and we know that there's an actual genetic difference between the M and Z flies in this rim allele. So we wanted to really confirm this tested further, so we got flies that are null for rim entirely. So Miller and colleagues published in 2012 on a stock of flies called Rim 103. These flies are missing this entire portion of the rim gene. So this removes 2 exons as well as a large part of the 3rd exon. It removes the entire zinc finger, CQA, and PBZ kinase from the rim protein. It's entirely non-functional. This copy of the protein does nothing. So flies that have this basically don't have rim. And so we can do the same thing we did with the deficiencies before, but with Rim 103 over a Z chromosome as opposed to an entire deficiency. So a targeted genetic null, these flies, if they have Rim 103, only have the Z allele of Rim. And so you find that again, balancer flies that are expressing a normal M copy of Rim over a Z copy of Rim behave like M flies. Rim 103 null flies behave like Z flies. So these flies only have the Z allele of Rim. That's the only thing we change in the genome of the flies, and suddenly they're behaving like Z flies. And again, very statistically significant. It's important to point out at this point that we tested this on multiple genetic backgrounds of Z lines, so different exothemal lines collected from different regions. And these results hold true across all of them. So this doesn't seem to be some sort of weird specific interaction. So this is great. We confirmed that this protein is involved. We confirmed a couple different ways that the gene is involved. And we know there are genetic differences. So what we're working on now is doing precise modification of the gene to put those M SNPs into Z flies and vice versa. So it turns out you can just do that with CRISPR. And you make these flies that are the 2 Z SNPs and otherwise M fly, and the 2 M SNPs and otherwise Z fly. These flies are actually done. We have them in the lab. We got them right before we came here. And we expect that these flies should recapitulate what we've seen in previous experiments. We expect these to behave like Z flies, and these to behave like M flies, based on what we found about them. So just to summarize what I've shown you today, melanogaster females show a very strong preference for cosmopolitan or Zimbabwe males, depending on what their own population is. The Rim gene is definitely involved in the CNN mate preference in Drosophila melanogaster females. And when we can do the targeted genetic modification of Rim, that will really elucidate the active genetic mechanisms driving this mate choice change in these populations of Drosophila melanogaster. So just to sum up, I'd like to thank everyone in the lab, Jane, my advisor, who's been just wonderful. I can't say his praises enough. Everyone else who helped, Jeffrey Liu, a former tech in the lab, and Graham Davis, whose lab we got the remote entry spots from. And with that, I'll be happy to take any questions. Yeah.
Do those computed positive steps
No, those are encoded region, and those are just sort of in the middle of the exon or first position of the codon. So they change coding sequence, but they don't change the actual splicing norms you get once the mRNA is processed.
Do you see any pleiotropic effects on other behavior in these in the Zimbabwe flies, and any suggestion as to what the behavioral mechanism is that leads to the mate choice preference?
Yeah, that's a great question. So not that we've been able to really quantitatively determine. Part of what's so interesting about this is no one's sure what the flies are discriminating against. They're phenotypically very similar. The F1 remake are perfectly fertile. So no, we haven't noticed. And the way you have to figure out if a fly is Z is to mate it to other flies. So no one's figured out really any other behaviors you can use to analyze them. Yes?
Is there any difference in the ecology of these flies that are made in different places? Or why is it Zimbabwe fly? Is that a unique biology or something?
That's a really good question that we're actually trying to answer. So there was just an expedition to Zimbabwe that collected over 300 flies, and it started hundreds of exophemal lines in the labs that are currently being evaluated. So that would answer that question. Yeah, we really like to know the answer as well.
So are either the Zimbabwe cosmopolitan populations polymorphic with those snakes we found in women? And if so, does that have any indicator of women populations?
So there is a small amount of polymorphism, but those sites are largely fixed. And if you look in lines that have polymorphism in other sites, that does not affect the nature of the behavior. So it doesn't. All right, thank you.